All right, so here in 15.4, we're going to talk about eukaryotic virulence, or eukaryotic pathogen virulence. The first one we're going to take a look at is fungal virulence. So, for example, Candida albicans, which is the causative agent for oral thrush, vaginal yeast infections, as well as cutaneous candidiasis. So this is our species that we're looking at that can cause all of those things. So let's take a look at some of the virulence factors that this particular organism produces. First of all, we have adhesins. So we've seen that again. Now we've seen that with bacteria. We've seen it with viruses. Here it is with fungus as well. So they have adhesins that are going to bind to the phospholipids of our epithelial cells and endothelial cells. So our epithelial cells that are inside of the body, so that's where we have the oral thrush and the vaginal yeast infection part, and then our epithelial cells on the surface of our skin would be the cutaneous candidiasis. Fungus or fungi also produce adhesins, just like we saw previously. Also, they produce proteases. So Candida albicans will produce a protease. So once it adhesins to the surface of the skin or the endothelial cell, and it's stuck there, then it'll start to produce these proteases or protease that degrades keratin. So if we're talking about the surface of the skin, for example, we have lots of keratin in our skin cells. As you get closer and closer to the surface or more superficially, there's more and more keratin, and that's a protective mechanism for our body, is to keep things out. It's a very hard protein that's within our cells. And so Candida albicans, what it can do is once it attaches to those cells, it can produce that protease, and then it can degrade that keratin. Once it degrades that keratin, then it can get inside of the body or inside of more of the deeper cells. Then it also has phospholipases. So phospholipases are going to break apart the phospholipids, and then that can affect the integrity of the host cell membranes. And then again, that's how it can get into the cells and can get through the cells into deeper tissues as well. We see the same with cryptococcus. Pneumonia and meningitis is from cryptococcus. In this case, they make a capsule, and that's what's going to make it more virulent versus our non-encapsulated cryptococcus. So we see a lot of the same things here with fungi as far as the adhesins, making proteases and lipases and creating or having a capsule in order to evade the immune system, making it more virulent and then allowing it to attach onto things and then invade the host cell. For Claviceps purpurea, it grows on rye and other grains, and this one can produce a toxin. So our Mycotoxin ergotism is from this. What happens is we have two different types of ergotism, which is gangrenous or convulsive. Our gangrenous is where this toxin, this mycotoxin, is going to cause vasoconstriction. So it's going to constrict or decrease the amount of blood that can go through the vessels, the blood vessels. Therefore, what it does is it ends up, because of this vasoconstriction, it ends up decreasing or completely stopping in some areas the blood flow to our extremities. So when this person is infected with this fungus and it produces this mycotoxin, then what it does is it's systemic, and so it'll cause all of the blood vessels to constrict, some more than others, and some so much that it actually stops the flow of blood, especially in the ones that are in our extremities, because those are smaller blood vessels. And then what can end up happening is we end up having death of tissue. So the fingers will actually start to die because it's not getting any blood flow. And it actually turns black and greenish because it's actually killing all of the cells in the body because it's no longer getting any blood to them, which is called gangrene. Same with the other extremities. So same thing with toes and the feet and things like that. Then the other type is convulsive. And so this was when the mycotoxin is going to then target the central nervous system. So rather than it being systemic in the bloodstream and causing this vasoconstriction, sometimes the toxin can go to the central nervous system. When it goes to the central nervous system, it can cause mania, which is kind of an extreme emotional instability, and hallucinations, so actually seeing things that aren't there. And so then this is the convulsive ergotism. Another example we have is Aspergillus. We actually looked at Aspergillus in the slides in the classroom. Aspergillus is a very common fungus. It is an opportunistic pathogen, Remember, what that means is that if a person already has a decreased immune system, say it's already fighting something off, then this Aspergillus can come along and actually cause infection because it has the opportunity to. Otherwise, typically our bodies are able to fight it off relatively easily, especially if we're just a healthy, well-fed, well-slept person. Oftentimes it enters via contaminated food or inhalation. So if there's fungus growing, the Aspergillus is growing around in the home or in a place of work or elsewhere, then the person can breathe it in, or if it gets on food, of course. Leads to aspergillosis. When a person has aspergillosis, they end up having a fever, as well as bloody sputum and or asthma, especially if it was through inhalation. So Aspergillus also produces mycotoxin. Its mycotoxin is called aflatoxin, and this is a mutagen, which means it can cause mutations in the host cells. It's also a carcinogen in the host, so it can cause cancer, and oftentimes it will cause liver cancer. So if somebody has aspergillosis and it starts to produce that mycotoxin, the aflatoxin, and it can actually cause liver cancer because it's a carcinogen, and then other mutations. So sometimes mutations do lead to cancer, but sometimes it's just mutations. This is also very dangerous because it does cross the blood placental barrier. So if a pregnant woman is exposed to Aspergillus, especially if they're already have a decreased immune system, not only just from being pregnant, for example, but also fighting something off, say it's autumn time and there's the flu going around, then they might actually have an infection with aspergillosis or have the aspergillosis infection, and then that can cross the blood placental barrier and be dangerous. There is a second mycotoxin, so gliotoxin, and what this does is it induces the host cells to self-destruct. So not only does it have aflatoxin, which can mutate cells or mutate the DNA and cause cancer, but gliotoxin can make the host cells themselves self-destruct. It also inhibits phagocytic cells, so it makes it so that the phagocytic cells aren't going to be doing their job anymore, so they're not going to be clearing the Aspergillus. And it's going to be part of the pro-inflammatory response. So it's going to cause inflammation. And again, inflammation is a good thing. Our body wants to have inflammation. That's why it does it, to help protect itself and get rid of pathogens. However, again, if we have a really high inflammatory response, then that can actually be dangerous to the host, to us as well. Also, we see Aspergillus has proteases like elastase. This is going to degrade elastin, which is the connective tissue. Where we see this oftentimes is in the lung, especially if it's due to inhalation. So if we inhale aspergillosis, it can then start to produce these proteases, specifically elastase, and that can degrade the elastic connective tissue that are in the lungs and cause lung disease. And then also catalase. So catalase is going to protect the fungus from hydrogen peroxide. Remember, catalase breaks down hydrogen peroxide. And that hydrogen peroxide is produced by the immune system. So sometimes when our immune system is going in and trying to kill an organism, it can utilize hydrogen peroxide. That's why we have peroxidases. We can utilize the hydrogen peroxide. It will be within the cell to get rid of the fungus or to try to kill the fungal cells. And then the fungus, like Aspergillus, can produce catalase in order to protect itself. So it can actually utilize that catalase to make sure that it doesn't get killed by our immune system. If we're talking about protozoa now, so we're moving from fungi to protozoa, we can talk about virulence factors for them as well. Again, some of the things that we've already seen. So adhesins, for example, toxins, for example, we see antigenic variation. We see the ability to survive inside of phagocytic vesicles. So we kind of saw that earlier as well when we saw bacteria that can get into the phagosome and then they can produce inside of the phagosome. And we see that same thing happening with protozoa as well. So a lot of these are similar. If we take a look at an example, we have Giardia lamblia, which produces giardiasis. And this is a large, or has a large adhesive microtubule disc. And so this really big microtubule disc is what's actually going to attach to the intestinal cells, to the mucosa. So if we remember back to other chapters or previous sections, remember that one single Giardia lamblia can cause infection. And this is because when this organism, Giardia lamblia, gets into the body, it has this big disc, and it's kind of like Velcro, like the rough part of Velcro, where it really can stick to the cells. So the flagella, the Giardia lamblia, if you look at the organism, it has flagella on it. It has this microtubule. It attaches to the intestinal mucosa. Then this flagella is moving, and it draws fluid from underneath the disc. And so there's this pressure difference. So as it's drawing the fluid from underneath the disc, it's going to cause that disc to press further and further and further into the cell. And that's what allows it to adhere to the cell. Once it does this, it causes inflammation. So our body is trying to respond to this and get rid of it, so it causes inflammation, but then it shortens its villi. And so when the cells are shortening their villi, that actually inhibits absorption of nutrients. So in our intestines, if we have our cells with our nucleus, remember we have villi in there, and these villi increase the surface area so that when nutrients are coming down our intestinal tract, we actually have more surface area with which to draw in those nutrients, including oxygen and releasing carbon dioxide, but then also bringing in all the nutrients from our food. So if we have Giardia lamblia is here and it's attached to the cells with its disc, so this is my Giardia, and then it's pulling fluid out from underneath there and it's sticking itself to the cells with this pressure difference, it causes inflammation, but then also is going to shorten these villi. Once it shortens the villi, if our villi now are down here, then we have much less surface area. And that's going to stop the absorption of nutrients or decrease the absorption of nutrients, which then causes a problem because not only is our body trying to fight off Giardia, what it's also doing is it's lacking the nutrients to do so because of the Giardia lamblia infection. Another example is Plasmodium falciparum. That is malaria, or the causative agent for malaria. Malaria gets inside of the red blood cells. What it does is it produces adhesins, just like we said. So it produces adhesins that move to the surface of the host cell. And what it does is it causes the cells to stick to each other and stick to vessels. So it kind of creates its blood clot utilizing these adhesins. So it's going to cause this clumping up of the blood cells. When it has a clumping up of the blood cells, then we have a decrease in blood flow. So if we have and that's because as it's doing this, right, as Plasmodium is sticking these cells together, clumping them together, not only are these cells being clumped together and therefore no longer useful as far as dropping off oxygen and picking up carbon dioxide, but they also will get stuck in our smaller capillaries. And as they're getting stuck in the smaller capillaries, then we lose blood flow to oftentimes the extremities and other areas that have small capillaries. Like think about our kidneys, for example. We have our little nephrons that are going to be filtering out our blood, and they're very, very small capillaries. So if we have these big chunks, this clumping of the blood cells, as that clump goes through, it can get stuck and it can stop the blood flow. And so then we can end up having things like organ failure and then ultimately death because we're cutting off the blood flow and we start to kill our own host cells. Plasmodium has the ability to go through antigenic variation. So this is one way that the body is unable to get rid of it. So our immune system is tricked every time because it has antigenic variation. Even if it thinks that it is killing the pathogen, it never is able to mount a big enough immune system response to completely clear that pathogen. And so this is why malaria is oftentimes a lifelong infection. It's a chronic malarial infection because it keeps changing what it looks like. Another example is Trypanosoma brucei, so the African sleeping sickness. In this case, what we have are capsules as well as antigenic variation. So again, those capsules are very, very dense glycoproteins. So it's similar to a bacterial capsule. So the immune system can recognize the way that it looks. So over time, maybe it'll recognize its capsule. But again, it makes it really, really big and hard to get rid of, hard for our body to clear, hard for our body to recognize. If it does recognize it, then we have antigenic variation. So the antigenic variation makes it look different every time anyway. Or maybe not every time, but a lot of times it'll make it look different. So even if our body is able to get around this capsule in this case, then our body is not able to clear it because it keeps changing what it looks like. So then if we look at some helminth virulence factors, what we have, again, we see similar things like proteases. So proteases that are going to degrade skin proteins like elastin, so elastase. We see this with S. mansoni. So when the organism gets into the body, when the helminth is in the body, then it's going to attach onto the skin on the body, meaning on the outside of the body, or even in our mucosal membranes. And then it'll start to degrade the elastin. So again, that's part of our connective tissue. So it can degrade the elastin so that it can get into the body. Now, a lot of our helminths are too large for the immune system. So our white blood cells, our phagocytic cells, are floating around there, and they don't have a problem typically with gobbling up bacteria. Bacteria tend to be smaller than our very small white blood cells. Same thing with viruses. So as it's going around, it can bring it into itself in a phagosome. But if we're talking about a white blood cell here, and then we're saying like this is what the size of a bacteria, this is the size of a virus, and we're looking at something like one of these worms in our intestines, our phagocytic cell is going to have a really hard time trying to gobble up that worm, right, that helminth. So first of all, too large. Secondly, some are protected by a tough outer cuticle. So some adult roundworms, so our trichinosis and ascariasis, they have this really tough outer cuticle that's kind of like the keratin in our skin, or you can think of it as kind of like nails, as in fingernails, on the outside of the helminth. And so that can be very, very difficult for our body to break down. It would take a lot of enzymes to do that. Then also, the surface characteristics change over the course of its life cycle. So if we take a look at any particular helminth, remember they go through these life cycles. And as they're going through the life cycles, the surface proteins end up looking different. And so even if our immune system is able to recognize that at some point in the life cycle, it's small enough to where phagocytic cells can go in, grab it up, and degrade it and alert the immune system, as that organism is going through its life cycle, not only is it getting bigger and bigger, not only is it producing this tough outer cuticle, if it's one of those organisms, but it's just changing its surface proteins in general. So it's making it so our immune system doesn't recognize it anymore. And if it doesn't, then of course it's just constantly initiating a first-time immune system response. And then lastly, we have our polysaccharides or glycans. So the polysaccharides or the glycans are on the surface of the helminth. We're still talking about helminths here. And then what happens is that they end up looking like immune system cells. So this is one way that a helminth kind of tricks our immune system. So we have several ways that we just spoke about, but then not only do they change the way that they look, they also change the way that they look to look like our own immune system cells. So they look like immune system cells and they can evade the immune system. This is called glycan gimmickry. And this is because they're making it look like the glycans that are on our cells. And so our body, when it comes along, it says, oh, no problem here. This is a self cell, because that's how they communicate, is by taking a look at these glycans on the surface. Then we have immune system suppression. So via these proteases, so breaking down the proteins, they can degrade antibodies. So via the proteases to degrade antibodies, for example, our S. mansoni, can decrease the immune system then. So not only is it difficult for our immune system sometimes to mount an attack, they actually have proteases that are going to degrade the antibodies themselves and break them down. Then we have other substances that also suppress the specific immune system cells. So they can also release large amounts of material to overwhelm the immune system. So not only can they do this thing where it makes it look like they're part of the immune system and then produce proteases to break down the immune system, but then they can also just put stuff out there that is not them, but is also not us. And so our body will go in and it'll start to attack all this stuff because it wants to get rid of it because it's not a self cell. And then that's making it so that our immune system is busy working on all of this other stuff while this helminth continues to grow and reproduce and continue infection.